Good morning, dear students. Welcome to the science class. Today we continue our discussion on chapter 2, microorganisms, friend and foe. In the previous classes, you have learned what are microorganisms, the classification of microorganisms, bacteria, fungi, algae, and protozoa, the characteristics of viruses, where do microorganisms live, friendly microorganisms, the useful microorganisms, use of making of curd and bread, commercial use, method, medicinal use, the use of increasing soil fertility, cleaning of the environment, the harmful microorganisms, the disease-causing microorganisms in humans, disease-causing microorganisms in animals, disease-causing microorganisms in plants, and food poisoning. In today's class, you will learn about food preservation. Microorganisms spoil our food. Spoiled food emits bad smell and has a bad taste and changed color. Food preservation is the technique to prevent food spoilage, food poisoning, and microbial contamination in the food. Food preservation is the technique to prevent food spoilage, food poisoning, and microbial contamination in the food. Let us study the common methods of preserving food in our homes. You have to save food from the attack of microorganisms. So this diagram shows the different methods of preserving food. Preservation by adding oil and vinegar, keeping the food in airtight containers, heat and cold heat treatment, preserved by adding sugar, preservation by adding common salt, the cold treatment. Let us see all these methods in detail. First method is chemical method. Salts and edible oils are the common chemicals generally used to check the growth of microorganisms. Therefore, they are called preservatives. Salts and edible oils are the common chemicals. They generally used to check the growth of microorganisms. Therefore, they are called preservatives. We add salt or acid preservatives to pickles to prevent the attack of microbes. Sodium benzoate and sodium metabisulfite are the common preservatives. Sodium benzoate and sodium metabisulfite are the common preservatives. They are also used in jams and squashes to check their spoilage. Second method is preservation by common salt. Common salt has been used to preserve meat and fish for ages. Meat and fish are covered with dry salt to check the growth of bacteria. Salting is also used to preserve amla, raw mangoes, tamarind, etc. Third method is preservation by sugar. Jams, jellies, and squashes are preserved by sugar. Sugar reduces the moisture content, which inhibits the growth of bacteria, which spoil the food. Fourth method is preservation by oil and vinegar. Use of oil and vinegar prevents spoilage of pickles because bacteria cannot live in such environment. Vegetables, fruits, fishes, and meat are often preserved by this method. Heat and cold treatment. Boiling kills many microorganisms. Similarly, we keep our food in the refrigerator. Therefore, low temperature inhibits the growth of microbes. Boiling kills many microorganisms and low temperature inhibits the growth of microbes. Milk is boiled before it is stored or used. Pasteurized milk can be consumed without boiling as it is free from harmful microbes. Let us see what is pasteurization. The milk is heated to about 700 degrees Celsius for 15 to 30 seconds and then suddenly chilled and stored. By doing so, it prevents the growth of microbes. This process was discovered by Louis Pasteur, so it is called pasteurization. Milk is heated to about 700 degrees Celsius for 15 to 30 seconds and then suddenly chilled and stored. So this is called pasteurization. The sixth method is storage and packing. Dry fruits and vegetables are sold in sealed airtight packets to prevent the attack of microbes. So the first method is chemical method, second method is preservation by common salt, third one is preservation by sugar, fourth one is preservation by oil and vinegar, fifth one is heat and cold treatment, sixth one is storage and packing. Now let us study nitrogen cycle. Our atmosphere has 78% of nitrogen gas. Nitrogen is one of the essential constituents of all living organisms as part of proteins, chlorophyll, nucleic acids, and vitamins. The atmospheric nitrogen cannot be taken directly by plants and animals. Certain bacteria and blue-green algae present in the soil fix nitrogen from the atmosphere and convert it into compounds of nitrogen. Once nitrogen is converted into these useful compounds, it can be utilized by plants from the soil through their root system. This you have already studied in chapter first. Nitrogen is then used for the synthesis of plant proteins and other compounds. Animals feeding on plants get these proteins and other nitrogen compounds. When plants and animals die, bacteria and fungi present in the soil convert the nitrogenous waste into nitrogenous compounds to be used by plants again. Certain other bacteria convert some part of them to nitrogen gas, which goes back into the atmosphere. As a result, the percentage of nitrogen in the atmosphere remains more or less constant. Let us see once again the atmospheric nitrogen, 78% of atmospheric nitrogen. So the lightning can fix atmospheric nitrogen into the soil. Nitrogen fixing bacteria and blue-green algae can fix atmospheric nitrogen into the soil. So they convert this nitrogen into nitrogenous compounds. So the nitrogenous compounds are present in the soil, and these nitrogenous compounds can easily be absorbed by plants. When plants and animals die, and the excreta of the animals are converted into nitrogenous compounds by the action of bacteria and fungi. These are used by plants again. And some other bacteria present on the soil, they will convert these nitrogenous compounds into nitrogen gas. And this will go back to the atmosphere. So this is nitrogen cycle. Dear students, this is all for today's class. The next class, we will discuss the questions and answers. Till then, bye. Take care.